Thank you. That was great. Now I'm going to pass over to Arjun to start his presentation. Arjun, over to you.
Hello, everybody. Thank you, Charlotte. Before I get into the topic of today's webinar, I just wanted to briefly talk about the center that I work for. We are the Golden Leaf Biomanufacturing Training and Education Center located in Raleigh, North Carolina. We are part of the NC State University. There are a lot of different things that we do here at BTEC with the main goal of education and training. We have a master's program. We also offer minor for undergraduates. And for professional industry professionals, we have multiple short courses, such as in downstream, upstream, analytical, as well as some special topics such as gene therapy and automation and so on. We also offer contract services. We have basically any companies or individuals who have ideas, don't have equipments, we typically execute projects for them. Um, Again, in all the fields such as upstream, downstream, analytical, and so on. We also get quite a bit of funding from different institutions, organizations, as well as NC State to do a lot of bioprocess research. That's how this particular gene therapy project has been progressing. The main focus of our center is on biological products for treatment and prevention of human diseases. These are some of the pictures of our facility and our lab space. And but if you were to take any of the short course, you would be doing using these instruments hands-on. And if you want to learn more, please visit our website. Or if you're in this area, contact us, and we are happy to show you the facility. Now, getting into the topic of today's webinar, I would like to briefly or initially talk about the research that I've been doing for the past few years, and then move on to tell you guys how in the recent past we have utilized PATfix to streamline our research process. My research was focused on developing serotype agnostic peptide ligands, affinity ligands for universal purification of AAVs from the cell culture fluids of HEC293 as well as the SF9 harvest. I work closely with an initiative at NC State called NC Viral. We work with different viral vectors such as lentivirus, adenovirus, adeno-associated virus, and so on. But the main focus of my study was as adeno-associated virus, which are basically the smallest viral vector, have the ability to encapsulate about 4.7 kilobases of single-stranded DNA. And they have a very nice property of tropism, because of which their preferred mode of delivery is in vivo. And some viral vectors are also delivered ex vivo, wherein the cells of the patients are taken, genes are transferred, and the modified cells are put back into the patients. Looking at the current state of art for AAV production process, there are multiple steps involved in upstream and downstream processes, all the way from the seed vial to bioreactor to different chromatography steps and TFF and bulk fill. The main focus of my work is on purification aspects, and I focus more on chromatography. On the right side, you can see there are a bunch of different resins that are currently in the market. Their reported binding serotypes, their approximate cost per liter, and their binding capacities, as well as some of their lifetimes. There are multiple challenges that the industry faces in designing the purification processes for these AAVs. The two most important ones that we targeted in our studies were the prone to loss of transduction activity upon long-term exposure to low pH. All the affinity resins that are currently in the market typically utilize low pH-based elution, due to which the integrity of the capsid or the structure of the capsid can change and come back to its normal state when neutralized, and all this could be detrimental to the capsid's transduction efficiency. And the second one is the high cost of these affinity resins that are in the market. So in order to do that, we started exploring some of the properties of AAV. One of the very interesting property of AAV, like we spoke about, is tropism. In order to show tropism, they have to go to a specific tissue and bind to it. In order to attach to these tissues, they attach to two different types of receptors. One is called primary, and the other one is secondary receptor. And the secondary receptor is the one that typically helps engulf the capsid into the tissues. And if you look on the right side, there are a bunch of receptors that are listed, but if you pay attention, there is a receptor called AAVR, which is the adeno-associated virus receptor, which is one of the receptor that most of the AAVs tend to bind to when they're attaching to the tissues. And this was very interesting for us. So we started doing some studies behind this interaction between AAV and the AAVR. And we did a bunch of in silico modeling studies, specifically molecular docking studies with AAV2 and AAVR. Uh, and then we discovered a bunch of peptide-based ligands that could have the ability to attach to AAVs while doing some purification work. We also explored another interesting molecule called A20, which is an anti-AAV mouse antibody, which has the ability to interact with intact capsids rather than the fragmented capsids. So we utilized these two molecules, two interactions, did a bunch of studies, and then figured out a sequence of multiple sequence of peptides that could have the potential to bind to AAVs, and which could be used for doing purification studies. The other interesting thing was they were all modeled in such a way that they could bind at neutral pH, and also the elution can happen close to neutral pH environment. So these were some of the peptides that we had discovered. I just wanted to show you guys that we also looked into their interaction with different AV serotypes. So all the colored cartoons in this slide are the different peptides that were discovered, and all the gray cartoons are the different VP1s of different AAV serotypes. It was interesting that the dissociation constant of these interactions at pH 6 was about 2 logs higher than that of at pH 7.4, which gave it a very nice little nudge to desorb AAVs from the column. So once we had discovered these peptides, we started synthesizing them on the resin beads, and then packing columns and doing some purification studies. So before we started challenging them with lysates, we wanted to make sure they can actually bind to AAVs. So we just ran some pure AAVs. First, obviously, was AAV2, which is what the discovery was performed on. On the left, you can see a nice little structure of AAV2, followed by a few products that are currently in the clinical trials based on AAV2. There is also an approved product in the market, Luxturna. It's a very popular serotype, a lot of studies going on in this particular area. And if you look at the histogram, that's the purification studies that we did. The first bar indicates the feed sample, and then we are seeing all the step yields from different peptides. I've listed only a couple of peptides that were more competitive to the ones that are to the AAVX and AVB resins that are currently in the market. So the number in red basically indicates the step yield. So you can see we got very nice step yields for different peptides. So once we knew that, yes, these peptides have the ability to interact with, that the peptides have the ability to interact with the AAVs, we wanted to challenge them with cell culture fluids. And then we started doing some purification with the HEK as well as the SF9 cell lysates. On the left side is the data for HEK cell lysate. You can see that again in the red are the step yields, and now in this case the blue numbers are basically the log reduction values for the host cell proteins. And you can see we did a pretty good job even when we are comparing it with the commercially available resins. Similarly, the same was the result for the SF9 cell culture fluids as well. We then wanted to make sure whatever we are doing is actually effective. And then we did a transduction assay with human epithelial cells, HT1080 cells, wherein we took the LUHs from the different peptides and then transduced the cells. And you can see the green bars on the histogram here indicate the 3 different peptides, that the LUHs that we tested, followed by the blue bar, which is the AAVX solution, which was neutralized after it came out of the column. And then the last one, the red red AAVX LU8, was the sample that was never neutralized. So all we wanted to figure out was, or we basically understood that keeping the capsids happy at neutral pH environment is the way to go and to keep the capsids happy here. We also wanted to look into the quality of the capsids just to make sure they are not fragmented or they are all intact, not forming aggregates and so on. We imaged them under imaged the eluates that came out from the column under the mini transmission electron microscope, and they all looked very well intact. We didn't see any big issues here. We also looked at the particle diameter, and they were all distributed between 25 to 30 nanometers in size, which is what we expect for a typical AAV diameter. Finally, we also looked into the dynamic binding capacities of these peptides. These peptides were attached to the Toyo Pearl resins, and then when we did the dynamic binding capacity, we challenged them with AAV2 in cell culture lysates, such as HEC293, and we found most of the peptides exceeded 2e to the 14 in the 10% breakthrough dynamic binding capacity per ml of resin. Very comparable to what we would see with the commercially available resins as well. And they also seem to maintain their binding capacity even post 20 cycles of usage. All the data that we saw until now were typically from different assays that were all plate-based assays, wherein you determine the concentration of a sample targeting a specific molecule. But what exactly is present in the whole sample is much more better visualized when you get some visual confirmation. So that's when we started integrating HPLC-based methods, and that's when we started looking into the integrating the PATfix platform here. This is an image of the system that we have here at BTEC. The very interesting thing about the system is its ability to have multiple detectors in line. So by just injecting one sample, you can get a lot of information about the sample. The nice thing in our system is we have UV, we have mols, we have fluorescence, we have refractive index detector as well. We can get multiple information through that. And we also have a fraction collector, if you look up there, which can, like if you're running critical process samples, if you want to collect it, we have that option as well. One very cool feature that I like about the system is it also has pH and conductivity sensors in line, which very well helps us when we are running gradients. You just don't rely on percentage A and percentage B to make assumption that the gradient is running fine. You can actually see that gradient forming by the change in conductivity traces and the pH traces. So we utilized the PATfix platform to run some methods that we have been working on. One was the size exclusion chromatography. We utilized an interesting 200 Armstrong pore sized column, wherein in a 200 Armstrong column, you will not see the AAV go through the pores. They will just flow through the void space, and you'll get a AAV peak initially, and then all the host cell proteins and impurities just go into the pore size and come out much later. So you'll have a pretty good baseline separation between the AAV peak and then the other impurities. So if you look at the first sample, in this case was the feed lysate sample. You can see a nice AVP coming out initially, and then you have multiple impurities. And then later on, when we ran the elution samples from different peptides, we got very nice single AV peak and tiny blips of impurities, which were still lingering there. But this clearly was the visual confirmation that I was talking about, wherein you can see the purification actually happening, right? So I'm pretty sure that the peptides are actually doing the job they're intended to do. We were using fluorescence detector in this case. I think I have the excitation and emission switched in this case. It was excitation at 280 and emission at 350. Also wanted to show you that it works across the board. We tested different serotypes. I did not present all the serotypes here today, but we worked with different serotypes. There was one of the peptide that acted as a universal binder, and we ran all the AAV serotypes through the columns, and we were able to get similar results with most of the serotypes. So just wanted to show you guys that it does not depend on the serotype. It works across the board. And you can see in the red here is the feed sample versus the blue traces are for the elution samples. You can see clear improvement in the or enrichment in the capsids. We also utilized in the recent times to quantitate the amount of capsids that are present using this method. You can do this by building standard curves with standard AAVs, and it works very well and has been very comparable to what we see with ELISA as well. The next method that we explored was the steric exclusion chromatography with the CMAC hydroxyl-based column. The concept of steric exclusion is very well explained by Pete Gagnon in some of his research articles. But basically, it's similar to a PEG precipitation technique wherein the larger molecules in this case tend to get retained back onto the column, and the small molecules such as the host cell protein and impurities just flow through, and you get two different peaks. Basically, if you look at this chromatogram here, the initial big peak on the first sample that is designated as A, you can see a big peak coming off initially, which is the host cell protein and impurities, and then you get a tiny peak of AAV. But when you run the elution samples, you can see clear enrichment or enhancement of those AAV peaks versus the impurity peaks. So clearly there are two peaks which are telling us if we have impurities or if we have reduced the amount of impurities and how much AAVs are still present in the column. So initially we started off with a 30-minute run, and we have now shortened these methods to about 7 to 11 minutes. So in theory, we can quickly get data off of a sample or information about a sample in about 7 to 11 minutes. Again, I would just wanted to show you guys that it works across the board. Different serotypes works. Everything can be tested on this particular method. One more interesting thing about this method is a student worked on this particular method. He was the one who actually designed the steric exclusion method. He injected different amounts of samples through the column and basically lysate samples, and designed a standard curve and got a very nice standard curve. So all I want to say is we can quantify the amount of AAVs in the feed sample, which is one of the most challenging things to do. If we do a plate-based assays, there are matrix effect and so on, but this assay can clearly give a baseline separation between the lysate as well as the AAVs, so you can build these standard curves and quantify the amount of AAVs as well. The next method that we are exploring, or we're still under developing this method, it was the ion exchange chromatography with the CMAC empty full. We are still just utilizing the method that was given to us by Sartorius BS Separations, and we have not completely separated the empty and full. We don't get the baseline separation yet, but the initial method itself is giving us a lot of information. This was for AAV8 sample. That is where the method was initially developed. On the left side, you can see the two chromatograms wherein we show the elution samples that were tested. We get clear separation of empty and full, and we can quantify the amount of fulls very nicely. And the one on the bottom is one of the standards that we tested. And we tested this with multiple orthogonal methods, and all of them seem to correlate well with each other. And we have gotten pretty good comparison, and we feel that this method is very reliable. And also looking into some of the initial presentation by Blaž, we can say that it is a very exciting method because they are developing this method to quantify the amount of percentages of fulls in the feed sample, which is again one of the most challenging things to do here. Finally, I'd just like to acknowledge all those people who have one way or the other helped me in this research work, as well as doing different purification techniques here. And then thank you so much.